Hello, my name is Harold Lee, and I am a field scientist with Pacific Biosciences covering the Central South region of the United States. And I would like to tell you today a little bit about our Kinnex kits and applications. There are three areas that I'd like to talk about. First, why do you need long reads for RNA sequencing? What are the Kinnex kits? And how do the Kinnex arrays work? So why do we need long reads for RNA sequencing? Not to put too fine a point on it, the main reason why you need long reads for RNA sequencing is because RNA molecules tend to be long. The average molecule size is on the order of 2,000 base pairs, and beyond that, somewhere on the order of 90% of all genes express multiple isoforms. And if you can't read through the various splice points in these isoforms, you don't know what isoforms the genes are expressing. Multiple isoforms create multiple proteins, and these different proteins have different functions. And over here on the right, I'm showing an example of the BCL gene where we have two isoforms. One isoform is pro-apoptotic and suppresses cancer. The other is anti-apoptotic and promotes cancer. If you were unable to identify the isoform that was being expressed, you would not know whether this BCL gene was promoting or suppressing cancer. More than that, when you are sequencing with the short read technologies, short reads tend to give reads on the order of a few hundred base pairs. A few hundred base pairs is insufficient to read through RNA molecules that are on the order of 2,000, 3,000, upwards to 10,000 and more bases in length. Beyond that, you're relatively unlikely to identify even a single splice site within any of the isoforms that were being expressed. Because of that, when you are done sequencing with the short read technology, you're left with a puzzle that doesn't have a unique answer because you have multiple reads. None of them are giving you a single full isoform. So you can take all of the reads and you can put them together in many different ways. Contrast that to long read sequencing, where we are able to sequence from the 3' region to the 5' region all in one read, and you get all of the various splice junctions from all of the isoforms in individual reads, and you get all of that information without having to do any assembly of the data. Now, when we're talking about RNA sequencing, there are a couple different ways you can think about RNA sequencing. You have bulk RNA sequencing, and then you have single cell RNA sequencing. Bulk RNA sequencing, we could think of like drinking a fruit smoothie. You know that there's kiwi and there's strawberry and there's banana in there, and you can kind of taste all of the different flavors, but all of those different flavors are all muddled together in one shake. Single cell sequencing is more like eating a fruit salad. You can see the kiwi, you can see the strawberry, you can see the blueberry, and each of those have been partitioned away from the other so that whenever you want to look at the data from one cell type, you can see that individual cell. All of the information from that cell type is assembled together and clustered so that you're only getting the information from, in my analogy, only the information from the kiwi or only the information from the banana without any confounding from the other fruits that are in this salad. So when you're doing RNA sequencing, there are a couple different goals that you have in mind. And regardless of bulk or single cell, you should be interested in the isoforms because, again, the isoforms are what are driving the biology. And you may or may not be interested in cell-specific information. But regardless of whether you are or you are not interested in the cell-specific information. You're not getting the isoform discovery with the short reads. It's only with the long reads that you're able to do the discovery and quantification of the isoforms that are actually driving the biology that you're interested in. And you're not getting that information with the short read technologies. So let's move from why to what. So what are the Kinnex kits and how are they distinguished from themselves and from other protocols and applications? So we have 3 Kinnex kits. We have the Kinnex full-length RNA kit. This is the kit that you would use for the fruit smoothie, for the bulk RNA sequencing. You take your tissue or organism of interest, you extract the RNA, and then you sequence all of the RNA from that tissue or that organism. And you can generate full-length transcripts from anywhere from 1 to 10 kilobases in length. We also have the single-cell RNA kit. This is our kit that was developed for the fruit salad. So we're going to use the 10x platform to sequester out individual cells. That generates the cDNA that is then put into our Kinnex array system to generate the actual reads that you care about. You can generate up to 40 million reads on our Sequel II system and up to 80 million reads with the Revio system. And then finally, we have a very specific kit for microbial 16S RNA sequencing. This, like the name implies, is for RNA for 16S sequencing. We have very high multiplex, and you can generate between 25 and 60 million reads depending upon the system that you are using. So now, how do the Kinnex arrays work? So there's the first step of the array process is cDNA generation. So we take our RNA molecules, we create cDNA, and when we're creating the cDNA, we add a handle that allows us to do the second step of the process, and that is to do a second step of PCR where we add a very specific Kinnex adapter to each end of each of our molecules. By adding these adapters, we're able to tile the individual reads into one much longer read. So in the array formation step, we take all of those individual cDNA molecules and we ligate them all together stepwise and thoughtfully, A's to A's, B's to B's, all the way down, and we create a very long molecule for sequencing from relatively short molecules. In the example of a single-cell sequencing, we're going to take molecules of about 1,000 base pairs in length. We take 16 of those, array them out, and where we had 1,000 base pair molecules, after the Kinnex concatenation and array formation, we have 16 kilobase pair molecules that are actually going on to sequencing. We are able to do multiplex at a couple different steps so that you get a lot of information from the individual molecules. So we take this long molecule, we can do the sequencing on either the Sequel II system or the Revio system. We have a very long read length, which improves the what we're calling the high-fi conversion rate. We're able to sequence around this molecule many, many, many times. Because we sequence this molecule many, many, many times, we get many, many looks of each of the long molecules, and subsequently each of the segment molecules that created this Kinnex array. That allows us a very high accuracy in identification of the reads and the transcripts that created them. After the sequencing is complete, we do have a end-to-end solution going all the way from the cDNA generation through the analysis. The smart analysis software within our SmartLink suite of applications will do all of the demultiplexing for the samples. It will break the segmented reads out of the Kinnex arrays. It does the primary extraction step, which removes all of the biologically irrelevant information and leaves you with just the biologically important bits of data. We do the isoform clustering. Once the clustering of all of the various isoforms is complete, we go on and map that back to a reference, and then the transcripts are classified for whether this is a known or an unknown isoform. You get all of the various plots and output files in industry standard formats. And then finally, you are able to do this analysis on a sample or essentially project basis. So you may have several samples in each of the samples that goes on to the instrument. You could either say, I want to see only sample A in this analysis, or I want to see sample A through X combined in this analysis. So regardless of how you want to do it, you can do that analysis. And all of this is bundled up, like I said, in our smart analysis suite of applications. And with that, that is everything that I have for the Kinnex kits and applications. If you have additional information, if you have additional questions, you can submit those at support@pacb.com. Thank you.